Hey guys, my name is Will Price and I'll be doing my presentation on the MPC and its role in heart failure. To start off, I would like to discuss the MPC or the mitochondria pyruvate carrier. We discussed MPC a while back when talking about the TCA cycle. The MPC is located within the inner membrane of the mitochondria, as you can see right here. And it is composed of two subunits, MPC1 and MPC2, which we'll get into later on. And before getting started, I do want to let y'all know that the ability for it to carry pyruvate is regulated by its abundance and post-translational modification, which I will go into more detail later on. To start off, this next slide is going to discuss pyruvate, which we have covered in detail in class, but I'm going to go over it here because there are some pathways that are important to know for this study. The first one being the TCA cycle, which we all know is when pyruvate has to go through the mitochondria pyruvate carrier first to enter into the mitochondria, but once it's in, it can be oxidized to acetyl-CoA to where it goes through the citric acid cycle, and it produces NADH and FADH, which can help produce ATP during oxidative phosphorylation. And another pathway that results from pyruvate is gluconeogenesis. And lastly, is going to be pyruvate being reduced to lactate in the cytosol, as you can see here. And this will actually produce less ATP, but can result in glycolysis being maintained at higher rates. So this is going to bring us to the study. They wanted to see if the abundance and activity of the MPC mediates the progression of heart failure. To do this, they needed to understand the mechanisms that regulate the abundance and activity of the MPC. And this is going to lead to the heart. Now, the heart consumes a lot of energy. Now, where does it get this energy? Well, it gets 80% of it from fatty acid oxidation. And during heart failure, its reliance on fatty acids diminishes, and so glucose uptake and glycolysis increases. But it's not enough. The reason being is pyruvate being reduced to lactate in the cytosol, because pyruvate is being handled differently by the failing heart. And this is also due to decreased expression of MPC. Now, this is actually reminiscent of the Warburg effect, which we discussed in chapter 14 and went over in class. And now for this study, they discovered similarities between the metabolic remodeling of cancer cells and the disease-induced hypertrophic heart that fails. And this is because decreased MPC expression for both resulted in lactate accumulation and increased glucose uptake. And this brings us to the downregulation of MPC1 and MPC2. And this was observed in failing human hearts and mouse models of pathological cardiac hypertrophy. They found that decreased MPC1 and 2 expression in adult mice using transgenic methods promoted hypertrophy. And this was supported by other studies, such as knockout of MPC1 and 2 in transgenic mice. Also, TCA cycle intermediates still accumulated. But why? Well, it may be due to the mice developing adaptive routes that supply carbon to the TCA cycle to maintain energy supply to the heart. This can be mediated by ketone bodies and beta oxidation, which replenishes acetyl-CoA to provide energy. And so here we're going to talk about other potential mechanistic explanations for why decreased MPC expression leads to cardiac dysfunction. You might think of MPC knockout hearts of not having the ability to satisfy the energy demand due to decreased transport and oxidation of pyruvate. But ATP abundance remains unchanged. in order for a failing heart to be a failing heart, it has to have a switch in fuel source from fatty acids to glucose and also be energy deficient. This means that metabolic remodeling does not have a direct effect on cardiac energy provision. So it has to mediate other pathways which result in energy loss. And this brings us to how alteration in MPC expression leads to post-translational protein modification. And some examples of this is variations in fumarate, as well as elevated O-glucoanoculation of proteins, and glucose shunting to hexosamine biosynthetic pathway, as well as it being diverted to the pentose phosphate pathway. And interestingly enough, when this happens with decreased MPC 1 and 2, NADPH increases, which is required for fatty acid synthesis and membrane synthesis required for myocardial growth. As you can see here, this is the picture I have, and that's where the knockout MPC or the alteration of the MPC is. And this is the elevated glucoanoculation, and this is the where the NADPH is increased. And this is basically like a updated version of the original that was shown in the, I think it was the pyruvate slide, or no, the MPC slide. So this leads us to rescuing of cardiac remodeling. Now, this can be tied back to a ketogenic diet, and this slide is going to discuss the benefits and negative effects of the ketogenic diet as it relates to the study. Now, a ketogenic diet can rescue the cardiac remodeling that occurs when MPC expression decreases. The reason being is that it impairs supply of glucose to cardiomyocytes from the liver. And they observed that chronic exposure of cardiomyocytes to beta-hydroxybutyrate, which is a ketone body, induced resistance to insulin. And this can limit supply to the pentose phosphate pathway and contribute to protection against cardiac remodeling. This also came with negative effects, which caused signs of systematic stress, dyslipidemia, and glucose intolerance. This led to a more targeted approach. They used a Dahl salt sensitive rat model of heart failure with preserved ejection fraction, which is the FPEF. And they used that instead of the original model, which had systolic heart failure. And they observed that expression of MPC1 increased, but MPC2 did not when exposed to 6 weeks of high salt. And this showed the difference between types of heart failure. But they found that ketone bodies protected against both forms of the cancers when administered in the diet. Unfortunately, there are still unanswered mechanisms behind this. But this can prove that ketone bodies are beneficial. And so this is going to lead us to MPC in ischemia reperfusion injury. And this is where MPC expression or activity in the heart might also alter cytosolic pH. And as discussed before, when decreased expression of MPC occurs, pyruvate is reduced to lactate in the cytosol. But we didn't talk about how it causes acidosis. And during myocardial ischemia, the pH is reduced to around 6.5, with acidosis being more marked because of this. And this can lead to intracellular sodium ions and calcium ions overloading, which causes swelling. And the acidosis occurring can be traced back to the accumulation of ketone bodies, which can lower blood pH, which we learned about in chapter 17. And so this is going to lead to the control of MPC expression and activity. Now, oxygen was discovered as maybe being an important determinant of MPC abundance. It was shown that MPC expression was strongly downregulated in the heart during late gestation, which results in delivery of oxygen being reduced. This brings us to when MPC expression is low and tumor growth occurs, which can promote hypoxia. And hypoxia can induce lactate accumulation and glycolytic flux by downregulating MPC levels. And this is actually going to bring us to how MPC expression can be regulated by transcriptional regulation, post-transcriptional regulation, post-translational regulation, and HIF1. Now this will bring us to transcriptional regulation of MPC. And this is downregulated by low oxygen levels. And the MPC1 gene, as you can see, has an established regulator called estrogen-related receptor alpha. And this can actually be activated by the peroxisome proliferator receptor gamma coactivator 1 alpha, which is the PGC1 alpha. And that is associated with upregulation of MPC. Now, where have we heard about the peroxisome proliferator receptor from? Well, we learned about this in class. It was a while back, and you can actually go to the slide, to slide 41 of fatty acid catabolism, and there's whole things about it. And they are also called PPARs, and they can stimulate the synthesis of enzymes required in beta oxidation when energy increase is demanded. And so after talking about transcriptional regulation of MPC expression, we're going to talk about HIF1, which is hypoxia-inducible factor 1 transcription factor. And this can induce pyruvate dehydrogenase kinase 1 expression to enhance the inhibitory phosphorylation of pyruvate dehydrogenase and repress mitochondrial utilization of pyruvate in the TCA cycle, which will result in hypertrophy, which is when the muscles in the walls of the heart actually thicken. And this will lead us to our next slide. So, in summary, the MPC is a way for pyruvate to enter the mitochondria, and we learned why this was important. And we also learned what controls its ability to carry pyruvate, which was its abundance and post-translational modification. We also learned about reduced or inhibited MPC and how that can be the result of lactate accumulation in the cytosol, which occurs from the uptake of glucose or increased uptake of glucose. And we also learned about the interventions of MPC activity. And so with all things concluded in this study, it is still hard to answer questions because of the complexities that surround this topic. But this study shows valuable insight into how MPC expression plays a role in heart failure. And so this leads to my first question. Why is the explanation of decreased transport and oxidation of pyruvate in the mitochondria not a solid answer to why decreased MPC expression leads to cardiac dysfunction? And this, for my question number 2, I have what is HIF1 and what role does it play in heart failure? You can go to slide 45 of chapter 14 for help if needed. Thank you.